Good morning, everybody. It's great to see everybody. Lots of familiar faces, and I hope everybody's well. It's a real pleasure for me to introduce Naz Mohammadzadeh. Naz is a PhD student in my lab, and will tell you about some work she's done on HIV persistence in the brain. Just by way of background, Naz is she's about halfway through her PhD. I was fortunate to recruit her from the University of Saskatchewan, where she worked with Linda Chelikos on APOBEC, and also was very productive there. So, and this project that you're going to hear about this morning is a collaborative project. It's part of the Canadian CURE project, so-called CanCure, led by Eric Cohen at the University of Montreal, but it really spans the entire country. So it's been a very exciting collaboration for everybody, and include particularly us. So I don't think I'll say much more except Naz, take it away.
Thanks, Chris. Hello, everyone. I'm Naz, and I'm doing my PhD with Dr. Power. So in the Power Lab, we are very interested in HIV-1 infection of the brain and associated neurological disorders. I'm going today to present to you some of my work on persistence of HIV-1 and SIV in the brain, despite effective antiretroviral therapy and neuroimmune activation. I'm just going to quickly start my laser pointer. Awesome. So we know that HIV-1 infects CD4 plus T cells and cells from myeloid lineage in blood and several tissues like lymph nodes, GI tract, lungs, and brain. Brain is seeded early during primary systemic infection. HIV-1 enters into the brain either in cell-free form or by hopping on the cells that can cross the blood-brain barrier. And in the brain, you can detect HIV in macrophages, in astrocytes, in microglia, and T cells. But neurons do not get infected. However, HIV-1 infection induces innate immune response and non-autonomous cell damage and death in neurons. I will be talking later on about microglia, so I will give you one slide refresher. Microglia are brain-resident macrophages that comprise 10 to 15% of brain cells. Besides their role in immunity, they have important role in development and homeostasis. They are CD4 positive and CCR5 positive, so it means that they support productive HIV-1 infection. Microglia are really interesting. They have long half-lives. They resist HIV-mediated cell death and cytopathic effects. So they're really important in terms of HIV persistence in the CNS, and they play an important role in associated neurological disorders and aging. So this is an image of microglia in resting mode. You can see the ramifications, which was labeled by IB1, which is microglia macrophage marker. And this is an image of HIV-infected microglia. So what are the consequences of HIV infection in the brain? Some of HIV-positive patients develop HIV-associated neurological disorder, or HAND, which manifests as declining memory, motor, and behavioral function. Pre-ART era, the proportion was 1 in 2 patients. Currently, 1 in 5 HIV-positive person develop HAND despite ART. Well, why is this happening? We know that there are several challenges for ART efficacy in the brain. Low penetration of ART across the blood-brain barrier, physiochemical properties of the drug, and different expression and function of transporter proteins on brain-specific cell types can impact intercellular concentration of these drugs in the brain. Nonetheless, suboptimal levels of ART in the brain can cause HIV drug-resistant mutations that have been reported in the polymerase gene. HIV-1 potentially can rebound and egress from the brain to the periphery due to these mutations or after ART interruption. The main question that we aim to answer, does HIV persist in the brain despite effective ART? We hypothesize that HIV has differential effects on viral RNA, DNA, and integrated DNA, and also immune responses in the brain. To investigate these, we want to assess ART effects on brain SIV and HIV-1 load in different treatment regimens, and also we want to compare efficacy of ART in the brain-specific cell type microglia compared to PBMC-derived lymphocytes. So our strategy is that we have two cohorts. We have a human cohort and non-human cohort. We use postmortem brains of HIV positive and negative patients and postmortem brains of SIV positive Chinese rhesus macaques to extract total RNA. This total RNA was used for cDNA synthesis and subsequent QRT-PCR to measure our genes of interest and droplet digital PCR to quantify viral RNA. We extracted total DNA from these tissues to quantify viral DNA and integrated DNA by droplet digital PCR. We also have fixed and paraffin embedded brains, which were sectioned and mountains and used for immunofluorescence imaging. So let's just start with the human cohort. In the human cohort, we have three groups: HIV negative, N of 3, HIV positive, N of 8. This group did not receive any ART at the time of death and had detectable plasma viral load and low CD4 T cell count at the time of death. We have 4 HIV positive patients that received ART and were suppressed. They were aviremic for at least 5 years, and they received ART by 12 hours before tissue collection. Then they were followed throughout their therapy. Their plasma viral load was undetectable at the time of death, and they had rescue CD4 T cell count. So we used these brain tissues and we wanted to analyze viral RNA, DNA, and integrated DNA. So we detected a virus gene throughout the cohort in HIV positive patients in the brain. What was interesting for us was that the HIV positive patients that were treated and were suppressed also had detectable plasma RNA, viral RNA, DNA, and integrated DNA in the brain. And the levels of RNA were lower in the treated and suppressed group compared to untreated group. However, it was not significant. It could be because of the lower sample size. The total DNA was not significant. However, the ART treated and suppressed group had higher integrated DNA compared to untreated group. So this was very interesting for us, but we know that our ART treated and suppressed group had receiving ART for a long time. So it was really important for us to see if we can find virus antigen in the brain. So we looked at the brain tissues, and we labeled them with IBA1, which is microglia macrophage marker, CD3 T cell marker, and HIVP24 capsid. And we found HIVP24 capsid co-localizing with microglia and T cell in both of our HIV positive cohort, regardless of the treatment status and outcome. So next, we looked at some immune responses in the brain. So I performed QRT-PCR and looked at a relative expression of these genes that we have selected. We looked at CD68, which is monocyte macrophage marker, CD3 T cell marker, and 4 interferon stimulated gene, as you can see here. So we saw that overall, our HIV positive untreated group had higher level of expression of these genes compared to the control. We see a modest reduction of these expression of these genes in HIV positive treated and suppressed group. However, the only statistical significant was MX2 that was higher in HIV positive compared to the control. So we wanted to look at all of our variables. We wanted to see if they have any correlation. We wanted to see a correlation between age, plasma viral load, CD4 T cell, HIV RNA, DNA, integrated DNA, and the relative expression of the immune response genes. So we plotted everything in this Spearman correlation metrics. So so this is basically all of our variables for HIV positive individuals. So the lower half of these metrics is the correlation coefficient. You can see the numbers, and the top half is the graphical representation. So the blue tinted ellipses show positive correlation. The red tinted ellipses show negative correlation. The boldness of the color and the shape show the strength of this correlation. The bolder the color and the narrower the shape means that there is a stronger correlation. Beside reassuring trends that we notice, for example, the negative association of plasma viral load and CD4 T cell count, one interesting thing that jumped out was a positive association of expression of CD3, which is T cell marker, and MX2, which is an interferon stimulated gene in the brain, which was very interesting for us because we also have seen co-localization of HIV-P24 in T cells in the brain. So this could be an indicative of role of T cells in antiretroviral response in the brain. So from this, we concluded that ART had suppressive effect on plasma viral load, but did not have any effect on brain viral load, and had modest effect on neuroimmune responses in the brain. We know that the human cohort, they received ART for a long time, they were infected for a long time, and we are not sure about the specifics of the ART regimen. So we needed to investigate this further using a more optimized model. So we're using SIV-infected Chinese rhesus macaques. And we want to answer how do contemporary ART drugs and their interruption modulate brain viral load in this cohort. So our Chinese rhesus macaque model, we have 3 groups. We have SIV positive, did not receive any ART, and they were sacrificed after they had detectable plasma and CSF viral load. Our second group, SIV positive, they received ART 4 days after infection. When they had suppressed plasma and CSF viral load, the therapy was interrupted, and the animals were sacrificed when they had detectable CSF and plasma viral load. Our last group, SIV positive, treated and suppressed, they received ART 4 days after infection. When they had undetectable CSF and plasma viral load, the animals were sacrificed. And the drug regimen, as you can see here, tenofovir, emtricitabine, raltegravir, oldartegravir, and ritonavir. So we first looked at 3 different regions in the brain. We looked at viral load in cortex, striatum, and cerebellum to see if there are any differences in terms of viral load based on the region. Overall, we noticed that there are highest incidence of viral detection, RNA, DNA, and integrated DNA in striatum, 41 positive detection out of 54 events, followed in striatum, followed by 31 positive detection out of 54 in cortex and 30 in cerebellum. However, a chi-square test of independence did not show that there's any correlation, and the viral load and the brain region were independent. So we pulled the data together, and we're looking at viral RNA, DNA, and integrated DNA in the brain of SIV positive Chinese rhesus macaques. And we're not observing any impact from ART treatment, suppression of the plasma viral load, or ART treatment on brain viral load in these monkeys. And this is really interesting because it is really good to just have this in mind, that CSF viral load is not a surrogate for brain viral load, because these group SIV positive ART treated and suppressed, they have undetectable plasma and CSF viral load. But as you can see, they have RNA, DNA, and integrated DNA in the brain. So we looked at the brain of these monkeys. We could detect SIV-P27 capsid co-localizing with macroglia macrophages, and in all of the groups, regardless of the therapy, treatment, status, and the outcome, and also co-localizing with T cells in SIV-positive untreated monkeys. Again, we are looking at all of our variables in SIV positive Chinese rhesus macaques. So the matrix is bigger because we're looking at all of these variables in 3 different regions: cortex, striatum, and cerebellum. I'm not gonna go through all of it. I'm just gonna point out 3 interesting things. What I found interesting at first was that all immune response genes at the bottom here, they're all positively correlated with each other, regardless of the anatomical site. Second interesting thing was that SIV RNA, total SIV RNA in the brain, and SIV RNA in the striatum were positively correlated with immune response genes, meaning perhaps these genes, induction of these genes and associated proteins can have an impact on reducing virus in the brain. And second interesting thing, viral DNA had a positive association with these genes that could have an impact in induction of these genes. But this is, again, just correlation metrics. You cannot read too much into it. To summarize the animal study, we found that ART and its interruption has little impact on brain viral load. So we want to know why this is happening. So for the next step, we're going to compare efficacy of the contemporary ART drugs in suppressing HIV replication in brain-specific cells, which was microglia, compared to PBMC-derived lymphocytes. So for this experiment, I isolated human primary fetal microglia and human adult lymphocytes and infected them with pseudotype virus. Cells were washed and incubated with different concentrations of these drugs. These are the same drugs that we use for the animal model and the drugs that are currently used in the clinic. The soup were harvested 6 days post-infection, and I used P24 ELISA assay to measure the P24, and this permit me to calculate EC50. So what we found was that the EC50 levels of the drugs, FTC, dolutegravir, ritonavir, and raltegravir, was higher in microglia compared to lymphocyte, which means that if all of these drugs cross the blood-brain barrier to the same extent, they have lower efficacy in microglia at suppressing HIV infection compared to what you expect their efficacy would be in lymphocytes. The only drug from the drugs that we tested had higher efficacy compared to lymphocyte was in microglia compared to lymphocyte was tenofovir. Overall, we found that HIV-1 DNA and RNA persist in the brain despite effective ART. We found that contemporary ART drugs have minimal effect on brain viral load, although they had really good effect on plasma and CSF viral load. And we found that ART interruption did not have any impact or any significant, basically, impact on brain viral load. And we found that ART drugs are selectively efficient depending on the HIV-infected cell type and the individual drug. So this brings us to the HIV persistence of the brain, this condition that we are seeing, and there we identified basically 4 players: ART, low blood-brain barrier penetrations, low CNS concentration, and specific physiochemical properties of the drugs that impacts their intracellular concentration, the immune response, lack of active antiviral response, which is a double-edged sword. You don't want a lot of immune activation because it can lead to neuronal damage and tissue damage. Cell type, we found that the drugs were less effective in microglia. This could be due to expression of higher expression of efflux genes in microglia. This is preliminary work that I have done. I didn't show the result, but I think it has been published as well. And also, we know that microglia resist HIV-induced cell death, and they are long-lived. And anatomical site could be a contributor. We are seeing more virus in the striatum. However, maybe due to our sample size, we are not seeing a statistically significant effect. So with that, I would like to thank my lab members and Dr. Power. Will did all of the IF imaging. Weston did all of the statistical analysis, medical microbiology and immunology department at U of A. And our collaborator, Dr. Stacker, helped us with the animal model. Dr. Angel, Gelman, and Gil provided the patient samples, and Dr. Cohen for his direction, and all our funding agencies. I can take any questions.
Great. Thank you. Again, another wonderful talk. Ron has his hand raised.
Yes, very interesting and very complimentary to the previous presentation. So thank you for that. I have a quick comment to which you might have, for which you might have some observations, and then a couple of questions. So maybe I'll just start with the comment, which is that despite all this accumulating data from your lab, from Howie's lab, from many, many other labs, general virologists continue to poo-poo the notion that the CNS is important as a reservoir for HIV. And I don't know what more to do about it. Maybe they're not reading our literature, and maybe you or Chris has, you Naz or Chris has some thoughts about that. But let me ask my questions. And so one is that, so you, I think it was the SIV brains that you saw higher concentrations of HIV provirus in people on ART, which is kind of paradoxical. And I'm wondering if, I may have misheard it, but I thought you said those people had HIV for longer, and that would give an opportunity for additional virus to accumulate and be archived over time. Do you have any information about whether this is archived provirus, or why is it higher?
Yeah, I cannot comment on the fact that they were infected for a longer period of time, that the HIV positive untreated group. I don't have that information. What we thought was that so throughout the therapy, HIV is gonna get suppressed, and as you mentioned, we're just thinking maybe it just being derived for getting latent, and it becomes just in form of the integrated virus. But again, it is a statistically significant, but we have 4 sample size, so
yeah,
I wish we had more of that.
My guess is the effect size must be very large given the p-value.
Yeah, it is in a log too, so yeah.
Well, okay. So the other, and this is a quicker question, which has to do with the potential for lower intracellular concentrations of the antiretrovirals, explaining the difference in IC50 for microglia versus lymphocytes. My guess is you're probably measuring extracellular concentrations of drug, and so if the microglia are pumping out the drugs more avidly than the lymphocytes, that might explain the difference in the IC50.
Personally, I have not investigated intracellular concentrations of the drug. We have a paper by Eugene Ashetop, I think 2017. He measured intracellular concentration of some of the drugs that some of them we are sharing some of these drugs here in microglia versus lymphocytes, and there were lower intracellular concentration of those drugs in microglia, and that could mean that there is higher extracellular concentration for sure. Of what I know for a fact from the same cells that I performed these experiments on, there is higher expression of efflux genes that basically pump out the drugs.
Very helpful, thanks.
Hi, really great
Hello.
talk. This is Janice Clements. I have a question because we do not find the same results that you do about SIV infection in the brain, but we do perfuse our animals. So we have no blood in the brain or in the tissues. And also for most of the time, we take them out very long on antiretrovirals. So they're on antiretrovirals for 6 to 8 months. How long were your animals on antiretrovirals?
Varying lengths of time until they were suppressed, but no more than a month, I would say. So your
They're
experiment
only on antiretrovirals for a month?
Like until they were getting suppressed, like the moment that were suppressed, they were sacrificed.
I see. So you
This
still have
was-
circulating cells in the brain then.
Yes,
That's
and
why
the-
your levels are high, because if you give antiretrovirals like you do in people for 6 months or 8 months, the RNA in the brain, you can detect it in the cells, but there's no free RNA, but it is latently.
yeah, for sure. And
Okay,
that's-
now I understand.
they
So
are
it's,
not perfused, and you mentioned that, and also we just wanted to have a short term infection and
right,
treatment to contrast the HIV cohort.
Yeah, so you're really looking at entry and how much it seeds the brain. Yeah, that makes sense.
And it is interesting because we started ART 4 days after infection.
Right, yeah.
But
We've
it's still
done-
there is.
Yeah, we've done that too. We've done both 4 days after, and then we've recently gone to later to see how that, going out 3 months will affect the level of the reservoir in the brain. So, but very interesting, and I'm glad to see you doing this work in monkeys as well as humans. It's
I'm a
very
big
exciting.
fan of papers are coming from your lab, so it's great talking to you.
Yeah, no, it's good. Very interesting work. Thank you.
And I want to conclude. Dr. Clement had a very good point for ART initiation, which brings us to the fact that pre-exposure prophylaxis is really important, at least in terms of reducing viral load in tissue reservoirs.
I didn't understand the question. I'm sorry.
Oh, there was no question. I would just say
Right.
like
Yeah.
even if
No, I
we
agree
start
with you, yeah.
ART 4 days after infection, if you're seeing robust
You still see virus in the brain.
yeah,
yes.
so
It goes-
pre-exposure prophylaxis is really important.
Right.
That's
Yeah, well, other people, not us, but other people, other labs have shown that in the rhesus macaque, that by 4 days you've established, the virus in all the tissues of the monkey. So that's, probably similar to people. We just don't get that opportunity in people.
To check them for this,
right,
definitely.
Okay, we are almost at the hour. So let me just close by first thanking our 2 trainee speakers today for 2 really excellent talks, as well as those who asked questions and attended. The questions were very thoughtfully addressed by our speakers. We will not have a conference next Wednesday. We are taking 1 week off, but in the chat, you will see something from the NIMH, where their Friday seminar series will occur next Friday, February the 4th, with Jonathan Karn, again, talking about viral latency in microglia. So the link for that is in the chat. If for whatever reason you don't have it after this, just drop us a note. We're happy to get that to you. So no talk next Wednesday, but there is an NIMH talk on Friday. Thanks, everyone.